Hey gang!
Welcome to our first ever vodcast on the mitochondrial electron transport chain and chemoosmosis.
Alrighty, so let's get right into it. The products and the reactants. So, the reactants of the electron transport chain are NADH, FADH2, O2, and an ADP molecule as well as a phosphate. The NADH and FADH2 come from the Krebs cycle. O2 waits at the end of the electron transport chain to accept electrons, and ADP and the phosphate are synthesized to make ATP. So then we have the products. The products of the electron transport chain are ATP, your dollar bill, and H2O. So you have water as a byproduct and ATP as your main product.
So let's get into the steps and categories of this very complex set of reactions. So first, we have NADH and FADH2 from the Krebs cycle. So they're the products of the Krebs cycle and the reactants of the electron transport chain, and they are oxidized and donate their electrons and their hydrogen ions to this process through a series of cytochrome proteins. So there are a couple different kinds of proteins that aid us in the electron transport chain. Number one is complex 1, which oxidizes NADH and transports its hydrogen ions to the intermembrane space from the mitochondrial matrix, and it begins bouncing electrons through the chain. Complex number 2 oxidizes FADH2 and continues bouncing electrons. Complex 3 transports the hydrogen ions from FADH2 through to the intermembrane space and continues bouncing electrons. Cytochrome C also continues this process. And complex 4 transports hydrogen ions to the intermembrane space and takes the final electrons from the chain and transports them to the waiting oxygen at the end to create water.
All right. So then, just kind of the second piece of this. So when NADH and FADH2 donate their electrons, they also lose their hydrogen ions. So this all occurs in the matrix. So the active transport proteins transfer the hydrogen ions into the intermembrane space from the matrix. This creates a large concentration of hydrogen ions in the intermembrane space, which forms a concentration gradient, and allows the hydrogen ions to want to flow back into the matrix, which it does through ATP synthase, which joins an ADP with a phosphate to make ATP. So this process, as a side note, is called chemoosmosis. So then at the end of the electron transport chain, oxygen, like Grace said, is waiting to receive the electrons that have been bounced through, after which it bonds to remaining hydrogen ions that are chilling in the matrix to create water. And then the FADH and NAD plus return to the Krebs cycle to be joined with H plus again to be reduced. So right here you can see what Grace talked about with the cytochrome complexes on the left. And then on the right you can just see right here how you have these hydrogen ions originally in the matrix pushed through with active transport, the concentration gradient up here flowing back into the matrix through ATP synthase. This right here also is another little visual. I found this really helpful when I was just looking at it as a process. It's a little more simple, but here we are if you want to copy it down.
So there are several stages of cellular respiration, and today we're just covering one of them. But to start off the process is glycolysis, then pyruvate oxidation within the mitochondria, the citric acid cycle, also known as the Krebs cycle, and here we are at the electron transport chain and chemoosmosis, which for our purposes we are condensing to one.
So the point of the electron transport chain in cellular respiration and photosynthesis is the same. It's to release energy from those bouncing electrons to be able to power the active transport proteins to build that hydrogen ion gradient to in turn produce ATP. So like I was saying, there are some differences between cell respiration and photosynthesis. First of all, in cell respiration, the ETC builds the gradient of hydrogen ions by pushing them outside, away from the matrix, because the cycle or the Krebs cycle happens inside the matrix. Whereas in photosynthesis, the electron transport chain builds the gradient by pushing hydrogen ions inside, because the cycle happens on the outside. So those are just two opposites right there. Additionally, in cell respiration, the electrons are used to connect to oxygen to create the byproduct of H2O. Whereas in photosynthesis, electrons connect at the end of the EDC with NADP plus to make NADPH. So the end goals of those are a little different. The cell respiration also uses NADPH and FADH2 to create ATP, whereas in photosynthesis, the ETC creates NADPH and ATP. Just as an overview, the electron transport chain is the ending reaction to cell respiration, whereas in photosynthesis, the ETC is the first step of the process. So these two boxes just show you some differences between what the electron transport chain does in photosynthesis versus cell respiration. Right here, you can see just how different it is in terms of in mitochondria in cell respiration. The H plus ions are broken off inside, right here in the matrix, pushed outside. And then in photosynthesis, you have the opposite. You have the hydrogen ions being pushed inside to flow back outside.
And then we're just going to do a quick look into the location of the ETC. So reactants are found to reduce within the mitochondrial matrix, and electrons bounce through the inner membrane and back into the matrix, while H plus actively is transported to create a gradient in the intermembrane space before chemiosmosis takes them back through the synthase into the matrix again, as you can see right about there.
There are actually many types of organisms that can use cellular respiration, as you can see from this helpful little graphic. So organisms from all kingdoms can use cellular respiration, heterotrophs and autotrophs, which is a common misconception. However, there are two types of autotrophs. One is photoautotrophs, and those are what we commonly think of when we think of plants. They use the sunlight to make their own food, whereas chemoautotrophs use chemicals, and they are fully capable of using cellular respiration. And heterotrophs, which require other substances to make their own food, also utilize cellular respiration.
And so without the electron transport chain, the products of the Krebs cycle would not be used in any productive manner towards energy. This ETC provides a place for these two molecules to be helpful, as they donate their negative electrons in order to build energy. And without the electron transport chain, ATP would not be created. And then in terms of our fun fact, only part of cellular respiration that uses the atmospheric oxygen, as well as 34 ATP for each glucose molecule is produced.
All right, so we're going to leave it at that, and we'll catch you tomorrow in class. But for now, that's it.
Good night, gang.